Listen to the steady rhythm of rain striking a solid surface. When this specific acoustic pattern enters an otherwise quiet room, it dictates the structural quality of human sleep. Clinical environments often treat soft rainfall as passive, noise-canceling static. The physiological data shows it is an active biological trigger. The standard assumption dictates that total silence is the strict requirement for deep, restorative sleep. To see why total silence often fails to optimize rest, we have to track a rhythmic sound wave from its entry at the ear down to its final chemical output in the bloodstream. This diagram illustrates a mechanism called neural resonance. This occurs when physical acoustic waves from the environment lock into phase with the brain's internal electrical waves. This exact phase alignment is the mechanical prerequisite for altering the architecture of deep sleep. Tracing this specific acoustic neural pathway shifts how clinical neurology approaches sensory interventions, turning sound into a targeted physiological tool. This 3D map shows the anatomical entry point for external acoustic signals. During sleep, auditory data travels first to the thalamus, located deep in the center of the brain. The thalamus acts as a sensory gate. It serves as the brain's primary nighttime mechanical evaluator. When it receives erratic, non-rhythmic environmental noise, the signals arrive as chaotic, unpredictable spikes. The gate actively filters these signals, blocking them entirely. Consistent, rhythmic acoustic patterns behave differently. Because their frequency is strictly predictable, the thalamus allows these steady signals to pass continuously through the gate. Once cleared, the thalamus projects them forward along a verified neural pathway directly into the prefrontal cortex. The immediate physiological outcome is a highly stabilized neural baseline across this region. The thalamus is a dynamic protector, actively maintaining cortical stability against environmental chaos. Within that stabilized prefrontal cortex, we find a localized electrical environment characterized initially by randomized firing patterns. As the continuous, rhythmic acoustic pulses arrive from the thalamus, they introduce a physical beat into the circuitry, initiating oscillatory entrainment. The external physical pulses force the internal neural firing to adapt to their exact timing. The cortical neurons abandon their sporadic activity and begin to fire in absolute unison with the acoustic pulses. This synchronized firing consolidates the localized electrical field into a singular, strong, low-frequency EEG delta wave. These specific delta waves form the structural architecture of non-REM sleep stages. By continuously mimicking and driving this electrical frequency, the acoustic rhythm structurally lengthens these restorative non-REM stages well beyond their baseline durations. Consistent acoustic rhythms act as a biological pacemaker, fundamentally remodeling our sleep architecture by strictly dictating internal electrical frequencies. That electrical synchronization in the brain also controls the systemic chemical pathways of the human body. Sustaining this biomimetic resonance triggers a distinct neurochemical cascade. The signal travels down from the synchronized prefrontal cortex through the neuroendocrine axis straight to the adrenal glands. This chart tracks the primary neuroendocrine output. As delta wave consistency increases, systemic cortisol production drops proportionally. High cortisol keeps the body in a state of heightened alert, which prevents deep neurological repair. Suppressing it creates a highly stabilized biological environment, defined strictly by low cortisol and high delta waves. Through this exact sequence, external sound waves actively alter internal blood chemistry. We can map this physiological circuit as a closed-loop system. It begins with rhythmic acoustic input clearing the thalamic sensory gate. This steady input forces oscillatory entrainment of delta waves across the prefrontal cortex. Prolonged delta activity then commands the endocrine system to lower cortisol. This precise physiological state enables the ultimate clinical outcome of restorative sleep, memory consolidation. The brain requires this exact combination, sustained delta waves functioning in a low cortisol environment, to effectively process short-term data and cement it into long-term memory. Relying on total silence provides no external structure for the brain to lock onto, leaving the timing and duration of these restorative cycles to chance. Active, targeted resonance ensures the cycle completes. This entire neurobiological output is downstream of a simple, rhythmic acoustic input. Environmental acoustics serve a strict mechanical function. Through the absolute laws of wave synchronization, they dictate our internal neuroendocrinology.